If I told you that every minute, if you took the DNA that you synthesized in your body every minute and stretched it end to end, it would reach from the Earth to the Moon and back. So this is an extraordinary amount of DNA. So actually, by the time you're 50 years old, you synthesized at least a light year of DNA. And that, if you think about what that means, that's light year is the distance that light travels in a year. That's, so it's an extraordinary amount of DNA that we make. And the fidelity comes in when we start to think about, for example, the mutations that cause cancer. We know there's hundreds of oncogenes and tumor suppressor genes that if you mutate will lead to cancer, but most people don't get cancer in their lifetime. So, this is kind of really an indication of how incredibly accurate these processes are. I think those things really are extraordinary when you think about it. I realized that there was this really interesting problem that hadn't really been thought about in detail, and it was the problem of how you begin DNA replication. So I think at the time a bit was known about how you actually do the copying of the DNA, but how you actually start that process turns out to be really interesting. And it's the point that everything's regulated at. So it's the point of all the regulation in the cell in terms of making sure that you replicate your genome exactly once. It has to start, but then it can't restart at that sequence, because if it restarted at the same sequence, you'd get extra bits of replication. So I think I realized at the time that there was something really interesting in trying to crack understanding how the process begins. This was a big question. You don't know what you're going to find. You've got a big question, but you really don't know where it's going to take you, and that's incredibly fun. I was born in Brooklyn. Did my childhood in the suburbs of New York, and I grew up wild, I guess. We had lots of freedom to run around and do whatever we wanted. It was the 1960s, and it was a very exciting time to be a kid, and it was a very exciting time to be thinking about science, because a lot of things were going on. I was particularly interested in the space race.
15 seconds.
I was 11 when the moon landing happened.
Mission sequence start. 6, 5.
We were on holiday in upstate New York, and we were in a caravan. We didn't have a television, so I actually listened to it in the front seat of my father's car on the radio.
We have a liftoff. Liftoff on Apollo 11.
When I look back on those days, it just felt like such an optimistic time, because I really felt like anything was possible when I was a kid.
4 forward, drift into the right level.
I was still sort of looking around for what I wanted to do, and I guess this was probably around 1976 or 77, and I took a genetics course, and this was when DNA was first really becoming a powerful explanation for genetics, and then I found this narrative to understanding life, that you could understand life through this molecule of DNA, that really captured me. Biology before this was a very boring subject. It was sort of a lot of facts you had to learn about how different orders of animals were related to each other, and it was very, very boring. But DNA sort of brought everything together, because it was that, together with evolution by natural selection, explains everything in biology pretty much, and so it felt like there was this narrative that was happening, and it was still very early on. We knew, I often show this little picture of a little rotating DNA molecule of 30 base pairs. Back then we couldn't even sequence those 30 base pairs. Now we get gigabases of sequence in no time. So it was very early on. We didn't know how development worked. We didn't know how transcription worked. We didn't know how promoters worked. We didn't know how anything worked. So it was kind of wide open to do what you wanted to do. I'm a product of Irish Catholic parents who, my dad came to the U.S. from Ireland as an adult. He and my mother were moderately religious, and I went to Catholic school until I was about 15 or 16. And it was sort of church every Sunday and all of this stuff. But very early on, I sort of quickly moved away from any kind of religious belief. But I think in a funny way that's had a mark on me, because although the answers were not satisfying, the questions were important. And the idea of asking big questions about why we exist and that, I think, did have sort of resonated with me and stayed with me in my life. It's also been a sort of a lifelong feeling of wanting to answer big questions and try, as a way really of connecting with the universe and connecting with humanity. I feel like I got something out of religion in a way. It is a picture of just DNA that had been isolated from an early Drosophila embryo. So early Drosophila embryos replicate their DNA incredibly fast. In like 3 minutes, they replicate their entire genome, whereas it takes us, 10 hours. And the way they do that is they initiate replication very, very frequently, so very close together. And so each origin only has to replicate a little bit of DNA. And so this electron micrograph really shows all of these replication intermediates in a very visual way. And I remember seeing that picture and thinking, I remember in that picture there's one particular bubble, replication bubble, that's very small, and it's sort of, I remember thinking, that's just, a few seconds earlier something was happening at that piece of DNA that we know nothing about. Just rewind that picture 5 seconds and something was happening at that spot. And that just felt like that was a really interesting question. Why doesn't it happen then again? At the time when I decided that it would be a good idea to try and understand how replication initiated, I think I kind of knew that this was going to be a difficult problem. And when I went to do my postdoc at Cold Spring Harbor with Bruce Stillman, started working on this. And I think it was when I started working on it that I realized just how difficult the problem was going to be. And I think it was at that point that I really started to realize that this was going to take decades to really get to the bottom of it. And so I often use this 30-year question as a way. I often tell people that it's a good idea to have a 30-year question, a 5-year question, and a 1-year question when you're thinking about science, because you have to start with stuff that you can actually do. Then you have to be a little bit more expansive, and then you have to have some end point that you're really trying to get to. I should say what I thought was really interesting about the problem. It wasn't just that I wanted to build it up and figure out how it worked. It was that there was something fundamentally really interesting in it, and maybe unprecedented in biology on some level, in that the system is built so that you have an event that happens, but then it doesn't re-happen. So it had this really interesting feature that you had to do it exactly once every cell cycle. And that was, I thought, going to give us something that really there wasn't a paradigm for in any other system at the time. The key thing that we found was that So in bacteria, replication initiation happens in one step. And so it has to be, bacteria go through all sorts of contortions not to re-replicate. And they only have one replication origin to deal with. What's happened in our lineage is that that initiation reaction has been split into two steps. And it's that two-step reaction that allows you to regulate lots of replication origins so that they only get activated once in a cell cycle. And to sort of put it simply, you do the first step at the beginning of the cell cycle, and you can do that at hundreds of thousands of sites in the genome, but then you can't start replication, and then you go into a second cell state where now you can start replication, but you can't do that first step anymore. So now all of those things that were put on the DNA that allow you to replicate from those sites, you can't put them back onto those sequences. So now you can activate thousands of replication origins in one cell cycle. So, now that we understand it, with most things, once you understand them, they all seem very simple, but at the time it was not so simple.
Did you feel that you'd learned something about your place in the universe?
Yeah, absolutely. Yeah. I mean, I feel like, what do I mean by place in the universe? I suppose I feel like I understand something about life. This is, when you learn about something in a textbook, you sort of understand it. But when you actually discover it, you get a much deeper sense of understanding. And in a sense, it also helps you understand how other things were understood. And so it connects you to everything in science in a way. You really feel, connected to the all life in a way, I think. Yeah. I guess that was always the goal. So, like I said, I'm a biochemist by training, and I'm interested ultimately in mechanisms. But I think, I think Richard Feynman had a quote that I often use in talks, which is, what I cannot build, I cannot understand. And I think that's really kind of the credo for biochemistry, which is that, it's fine to be able to take a cell that does something and take something away and show that that thing doesn't happen. It's a different thing to say, right, now I'm going to take all the things that I think are involved in this process and I'm going to put them together and I'm going to make that process happen without the rest of the cell. Because I think that tells you that you really understand it. If you can make it happen like it happens in the cell with a defined set of factors, then you really understand it. And so that's why we set out to do this.
Were there any times when you thought this is really just a crazy enterprise?
Yeah, most days I thought, I think the key was to try and I think if you go into something like this and you think the end point is to reconstitute the whole thing, you're almost certainly going to come a cropper because it's just not going to happen. That's too difficult. You have to find a way of breaking every problem into solvable problems, and so the two steps helped a lot because we could break that then into two steps, do the first step first and take the product of the first step as the substrate for the second step. That was very helpful. But then, especially in the second step, it was, I really tried to kind of take different bits of it and have different people have projects based around individual proteins so that they would get something out of learning something about those proteins without necessarily having reconstituted the whole thing. And then at the same time had a great postdoc named Joe Yields who then was sort of like the sweeper who took all this stuff and put it all together and got it to work. It was definitely not just another day in the lab. It was a pretty extraordinary day, and it was, you know, to kind of paint a picture of what it was like. Well, the first bit of, so the idea was to try and get replication to actually happen. And the way you do that is that you put a radioactively labeled nucleotide in, which is the building block for replication, and you look for its incorporation into DNA. And if it worked perfectly, we would have replicated an entire plasmid, a big DNA molecule. But the first bit of replication we saw was just a tiny little small product of DNA replication. But what was great about it was that it passed all of the tests to know that it was really a specific initiation event. It required all the things we knew it should require, and so we knew that it was the right thing. I think I just was very pleased. Yeah, you know, didn't go whooping around the lab, but was just really, it was a great moment. Satisfying, I guess, is the word.
Did you reflect on this idea that you'd set out decades earlier to do this?
Not really. I mean, I have to say, you know, I tend to think about the future and not the past very much. So I didn't reflect a lot. Just went forward and move on to the next thing. Feels great. I mean, it's sort of full circle moments in a way of kind of coming back to the lab bench and doing things myself. I've been about 6 months now doing it, and I've got my own project, and I'm very interested in it.
You're asking a 30-year question still.
Well, I probably shouldn't be asking 30-year questions at this point in my life, but you can't stop, I suppose, because you never get to the end. So, you know, it's good to still have some, you know, big things off in the distance, even though you might not get there. What we've come to realize is that yeast is one organism in all of eukaryotes, and we need to really understand more about the evolution of this process in order to really understand how it works. So a major project in the lab is trying to reconstitute this all with human proteins, and we last year had a nice breakthrough where we were able to reconstitute that first step with human proteins, and now we're working on the second step, sort of very analogous to what we did with yeast. But I'm sort of going back and asking more evolutionary questions about how the whole process evolved in the first place. And I'm using yeast genetics to do that, which is fun, and I'm having a great time. And the questions are really interesting, and I think there'll be interesting things coming out in the future.